The genetic code and why it's called the code of life. Nucleic acids are very complicated molecules, but their structure shows certain regularities. They are constructed from a limited amount of smaller building blocks. If we compare nucleic acid with a language, we can think of the building block as the letters of the alphabet of the language. With this analog, we say that the language of nucleic acid in the cell describe our inherited trait. It tells us if our eyes and those of our children are blue or brown, if we are healthy or sick. A brown-eyed child receives from its parent nucleic acid, which have the ability to direct the formation of protein required for the synthesis of dark pigment of the eye. It's the chemical structure of the nucleic acid which determine the chemical structure of the protein. The alphabet of the nucleic acid dictate the alphabet of protein. The genetic code in the dictionary which give us the translation of one alphabet into the. When the hieroglyphs were discovered, the archaeologists made use of Rosetta Stone, which contained the same alphabet written with Greek and Egyptian letters. In theory, one could use the same methods for deciphering the genetic code by comparing the chemical structure of a particular nucleic acid with that of the corresponding protein, letter by letter. However, it is not possible for technical reasons. Very simple and ingenious solution realized by Biochemist had a decisive advantage over the archaeologists since he could construct in the test tube a system which uses nucleic acid as a template for the formation of a protein. Such a system can be compared with the translation machine which fed by the scientist with a sentence written in the alphabet of nucleic acid. Then machine then translated the sentence into the protein alphabet. The machine then translates the sentence into the protein. What is the mechanism of the translation of the code within the cell? How we know what we know about genetics? In genetics, during the fall of 1868, a young Swiss physician by the name of Friedrich Miescher isolated a new type of compound from cell nuclei. He called this compound nuclein. Today we call it nucleic acid. 2 years earlier, George Mendel in the town of Brno finished a series of experiments which eventually turned out to be closely connected with Miescher's discovery. From very simple experiment with bee, Mendel discovered that our inheritance is packaged into many independent genes. Mendel's work marked the beginning of genetics as a science. Nucleic acid and gene, originally two widely separated concepts, together from the basis. Miescher's results were published in extenso only after his death in 1890. Mendel reported his observation in the first publication in 1866, which received little attention and was soon forgotten. For a long time, no connection between genes and nucleic acid could be seen. Using fruit flies as a model organism, Thomas Hunt Morgan and his group at Columbia University showed that genes strung on chromosomes are the unit of heredity. In so doing, demonstrated that genes are made up of nucleic acid. Every discovery marked the beginning of the new branch of science which has come to be called molecular biology, and which up to the present time has been concerned mainly with the biochemistry of genetics. Is concerned primarily with understanding biological properties that are transmitted from parent to offspring. The subject matter of genetics include heredity, the molecular nature of the genetic material, the ways in which genes, which determine the characteristic organism, control life function, and the distribution and behavior of genes in population. Genetics is central to biology because gene activity underlies all life processes, from cell structure function to protection. Learning what genes are, how genes are transmitted from generation to generation, how genes are expressed, how gene expression is regulated in the focus of this course. The important principle and concept are present